Hello, and welcome to this webinar, Innovative Advances in Neuroscience. My name is Megan Thomas, and I'm the Multimedia Editor for DDW and your host for today's webinar, which is being sponsored by ACROBiosystems. This webinar will cover how advanced technologies are proving beneficial to drug discovery and development, the current unmet need within drug discovery and development for central nervous system disorders, and where the opportunities and scientific breakthroughs are for new drug development. You will hear from Fiona Elwood, Janssen, Jenny Barnett, Monument Therapeutics, as well as Rosanna Zhang, ACROBiosystems. Dr. Fiona Elwood is the disease area Stronghold lead, Neurodegeneration, at the Janssen pharmaceutical companies of Johnson & Johnson. She heads the global J&J team seeking new therapies for Alzheimer's disease and other neurodegenerative disorders and drives development of a leading global pipeline of innovative treatments to ease the burden and suffering caused by common nervous system conditions. Over to you, Fiona.
Hello, and thank you. My name's Fiona Elwood, and I'm the Vice President and Disease Area Stronghold Leader in Neurodegeneration at Janssen, the pharmaceutical companies of Johnson & Johnson. There's been significant progress in science and medicine over the last 2 decades, but the impact and the burden of Alzheimer's disease on patients, on families, and on society remains. The World Health Organization estimates that 60 to 80% of dementia cases are due to Alzheimer's disease, which affects an estimated 55 million people worldwide. The number of people affected with Alzheimer's disease is growing. There's a huge burden, not just on society and healthcare systems, but also on families and caregivers. And in the US alone, it's estimated that the spend on looking after patients with Alzheimer's disease exceeds $320 billion annually. Because of advances in biomarkers and our ability to actually measure changes in the brain, we know that actually the Alzheimer's disease pathology starts 10, 20 years before people actually start to present with cognitive decline and dementia. So that the first step is that amyloid accumulates in the brain, followed by increases in a protein called tau, which aggregates inside of neurons. And then this subsequently progresses to cognitive decline and diagnosis. So actually, with those patients who have that preclinical Alzheimer's, amyloid and tau in their brain, but haven't yet experienced symptoms, it's predicted there are around 315 million people globally who suffer from preclinical as well as clinical Alzheimer's disease. So when we as a field have really invested decades in trying to understand the disease and develop meaningful therapies, we've actually come up against many challenges. And these are really, I would say, grounded in the heterogeneity of the disease itself. So Alzheimer's disease starts before we know it's starting. And in those patients who develop disease, there's significant heterogeneity from one patient to another. For example, with one of Janssen's clinical trials that was run around 20 years ago, after the fact, we learned that 20 to 30% of those patients didn't actually have Alzheimer's disease, but had other forms of dementia. So unless you can really identify the patients who've got those biological, pathological changes in their brain that you're targeting, that results in insufficient target engagement, enrolling the wrong type of patients. Also, with many of our therapies, we want to target the patients early in the disease because we want to preserve the function and the integrity of the brain for as long as possible. So previously, we had a lot of problem because the pathology was changing in the brain before patients developed symptoms, and enrolling those patients early in disease was burdensome and time-consuming, even if we could do it at all. And then finally, identifying a homogeneous population who are going to progress at a similar rate was also previously impossible. For example, you might have a patient who had mild cognitive impairment, and the question would be how rapidly would they convert to full Alzheimer's disease or full Alzheimer's disease symptomatology. For some patients, this could be within 1 year. For other patients, it could be within 5 years. If you study all of those patients together in a clinical trial, you're going to have a lot of variability and have to really increase your sample size in order to run a robust and statistically meaningful trial. But in the last 10, 15 years, there have been some significant advances that we've really taken advantage of at Janssen. So our strategy has been to apply these latest advances in precision medicine, data science, and digital health to identify specific and unique biomarkers, to identify Alzheimer's disease patients early, to discriminate Alzheimer's disease patients from other causes of dementia, to be able to track the pathology of disease in patients' brains with things like our tau PET imaging studies, and then to be able to use the latest in data science and digital health to reduce the burden on patients and to be able to take frequent and rapid measurements to identify progression in clinical signs and symptoms. So for example, when it comes to plasma biomarkers, we've developed and evaluated a blood-based biomarker that measures this specific protein, phospho-217 tau, in the blood of Alzheimer's disease patients. This is a simple way of identifying the right patients, Alzheimer's only and Alzheimer's with confirmed pathology at a specific stage of disease for our clinical trials. We've also deeply invested in PET imaging, specifically tau PET imaging, and have developed a novel method, not just to identify where in the brain the pathology sits, but we can measure how that spreads from one brain region to another. We can use this for patient stratification at the beginning of our clinical trials to make sure we're enrolling patients who've got just the right levels of pathology. But we also use it to measure the progression of tau pathology in the brain, which is useful as an intermediate marker of disease progression to help inform the decision-making in our clinical trials. And then finally, we partnered with several tech companies to really apply the latest in digital biomarkers for our trials. So for example, we partnered with one tech company that has a speech measurement, and it uses a recorded conversation on the phone that can be done by the patient in the comfort of their own homes and as frequently as we need to measure and record specific features of speech, including use of words, pauses between words, fluency in conversation. And then they use an AI ML algorithm to extract these differences in speech patterns and to identify how that correlates with specific Alzheimer's disease pathology and can predict the level and stage of a patient's Alzheimer's disease. At a recent Congress, we presented over a dozen abstracts on our plasma 217 tau assay, comparing the performance of this assay against established Alzheimer's disease biomarkers in different patient populations. It's able to detect phospho-217 tau very sensitively in the blood of patients, even before the appearance of cognitive decline. The performance of the assay is excellent, and it can predict the status of a patient's brain by 85% or better. So we can know whether the patient has amyloid in their brain, and we also know that as the phospho-217 tau assay goes up, so the level of tau pathology in a patient's brain has also gone up. We've applied this to one of our clinical trials as a pre-screening approach. So patients come in, we do a simple blood biomarker test to identify whether they would be candidates for the specific enrollment criteria that include a more time-consuming and costly tau PET screen. By applying this to the clinical trial, we've been able to reduce the screen failure rate. So previously, only about 30% of patients would have been positive with our tau PET screening. After filtering through this plasma 217 assay, our success rate is now as high as 85%. And that's reduced the cost of screening patients to enroll for this clinical trial by around 60%. So to close, I'm really proud to tell you about some of the advances that we've made at Janssen, building on our 60-year legacy in neuroscience by really harnessing this precision medicine revolution to reduce the burden, disability, and devastation caused by this condition. We believe that using these biomarkers is actually going to lead to many more treatment opportunities for patients, targeting the right molecule to the right patient at the right time. And in some cases, that right time is even actually being able to diagnose the disease before the symptoms have set in. We wouldn't have been able to do this on our own. The advances in Alzheimer's disease have been because of significant collaborations across the industry, along with academic and public-private partnerships. And that shared vision and that goal to treat the unmet needs for this huge patient population are what drive us to really deliver important therapeutics for the Alzheimer's disease patient. Thank you very much.
Thank you, Fiona. Next up, we'll hear from Dr. Jenny Barnett, who was Chief Scientific Officer at Cambridge Cognition, where she led clinical trial, healthcare, and R&D functions before spinning out Monument Therapeutics. She is the author of more than 70 papers, 6 patents, and 2 popular science books, and is an honorary member of the University of Cambridge Department of Psychiatry. Over to you, Jenny.
Thank you very much. I'm Jenny Barnett, CEO of Monument Therapeutics, and I'm going to be talking about how Monument is bringing precision medicine to neuroscience. So first of all, why do we need to do this? Well, you may be aware that brain disorders, psychiatric and neurological disorders in particular, remain among the greatest unmet medical needs, so both in terms of their prevalence and also the impact they have in patients' lives, the disability they cause, and the economic cost associated with that. The major difficulty here is that it's been very difficult historically to successfully develop neuroscience drugs. So around 90 or 95% of drugs in this area fail during clinical development, which of course disincentivizes pharma companies from working in this area in particular. Moreover, where there are drugs available in areas like depression, actually the approved drugs only tend to work for around half of patients. So they have efficacy that doesn't work for everyone, and they typically come with side effects that mean that a lot of patients don't enjoy taking these drugs. And as a result of those two things, low efficacy and side effects, the overall fact is that most patients don't take their prescribed drugs according to the instructions. So there's an estimate that around a third of patients take their drugs according to prescription for psychotropics, for example. So a great big gap to be filled. At Monument, we believe that we can use digital biomarkers to help solve this problem by getting the right patient on the right drug at the right time. We're doing this in the first instance by using them to accelerate and de-risk new drug development, but we ultimately see these as being something that can be used in clinical practice to reduce the trial and error prescribing, which is what psychiatrists in particular typically have to do, and ultimately to enable a real new era of precision medicine in neuroscience with things like dose titration and adherence monitoring and value-based reimbursement, all enabled through these digital biomarkers. In terms of the technology, we lean heavily on Cambridge Cognition's digital biomarker platform, which is called CANTAB. This is a platform that has been developed over a period of around 30 years, originating in the University of Cambridge and then spun out through a number of companies to Cambridge Cognition, from whom Monument spun out in 2021. The platform is a very well validated software base. It contains a menu of around 25 digital biomarkers that have together been validated in around 3,000 academic publications. They've also been widely used in clinical trials, so they're familiar to pharma companies and regulators, and around 1 million patient assessments have been completed at the last count. The thing that's really nice about these assessments is that they're highly scalable, so they don't typically involve any requirements for a particular language or cultural knowledge. They can be completed in a few minutes with no training or equipment anywhere in the world. As you can see here, they can be run on basically any digital device. What Monument has brought to that is a data analysis strategy that allows us to make judgments about the individual patient's performance on this test and use that to make inferences about brain pathology at the N of 1 level. We have an exclusive license to use Cambridge Cognition's platform in this way for patient stratification or combination diagnostic purposes, and this allows highly accurate assessment of the individual patient for enrolment into clinical trials and ultimately at the clinic level. So these biomarkers and the data analysis that we use give us some strong IP supported by multiple patents, and we also have IP around the drugs and drug formulations that we match with the patients. I'll go on and tell you about that now. So the thesis is really this, that we believe that many, if not most, CNS drugs have failed historically, not because the drug wasn't useful, but because the diagnostic criteria that we use to enroll patients into clinical trials, the diagnostic categories, do not reflect a real biological reality of differences between patients. So in any trial of depression, for example, there might be multiple people who all show low mood, but who have very different biological underpinnings to that symptom. The idea with Monument is that we use our biomarkers to segregate out homogeneous subgroups of patients who, whatever else is going on in their brain, at least share a common abnormality in a specific brain process. And we then use novel drugs reformulated for better efficacy and safety in order to remediate the underlying biological mechanism that drives that brain process and that brain abnormality. And by using this segregation with biomarkers, we believe that we should have a more homogeneous group of patients who are all likely to show the same response to any given drug. Our lead program is aimed at cognitive impairment associated with schizophrenia. If you're not familiar, schizophrenia affects around 1% of the population. And while there are many drugs available to help with the psychotic symptoms, so the hallucinations and delusions that typically result in the diagnosis, in around 3/4 of patients, what is left is major disabling cognitive impairments in areas like memory and attention. These affect around 3/4 of patients, and they can really influence patients' quality of life. So, for example, preventing them from continuing an education, holding down employment, having positive relationships with their social peers and their family, and leading also to greater healthcare utilisation. Unfortunately, there are no drugs currently approved for this condition, even though it's estimated to be a very valuable market, and pharmaceutical companies have historically tried to develop drugs in this area. This is one where we think this problem of heterogeneity has really been key in preventing these drugs from succeeding in the late stage clinical trials. This is not something that affects schizophrenia alone. The brain abnormality and drug that we are intending to use to treat that would also be relevant probably in a subgroup of autism spectrum disorders, which is about 2% of the population, and particularly in pediatric ADHD, which around 10% of school-age children end up diagnosed with. So these are cross-diagnostic issues that can be addressed, producing an opportunity for large label extensions. Our solution for cognitive impairment in schizophrenia combines, as all our programs do, a biomarker that defines the patient population and a drug that treats it. The digital biomarker here you can think of as being a measure of your ability to filter out irrelevant information in the world around you so that you can pay attention to the thing that you're trying to pay attention to, hopefully my voice at the moment. In schizophrenia, our biomarker picks up about 2/3 of patients as suffering from too weak an attentional filter and about 1/3 of patients as having a normal or even too strong attentional filter. And we use that biomarker to segregate out who would be suitable for treatment with a particular mechanism of action. The mechanism of action that we're using here is nicotinic agonism. Our specific therapeutic is a fixed dose combination of 2 nicotinic agonists. The reason for the fixed dose is that we get a better safety profile than either alone, so it overcomes some of the off-target gastro side effects that have previously shut down programs using nicotine agonists in psychiatric patient groups. It also provides better efficacy than either alone. In fact, we've done some preclinical work in the last couple of years demonstrating that if you optimize the ratio of drug A to drug B, you can get a really nice boost in cognitive enhancement. So we have data showing the synergistic effect of the drug combination in improving cognition preclinically. We also have both preclinical and clinical data demonstrating a strong mechanistic link between the biomarker and the drug. And in some very exciting brand new news, we've just found out that this biomarker and drug combination have been selected for inclusion in a major non-profit funded clinical trial due to start late next year. We're currently preparing to open the IND and preparing drug formulation and so forth. We understand that this clinical study, where we're still involved in defining the protocol at this point, but it looks like it will be around 120 patients for around an 8-week study with results reading out early in 2026. So really a major validation of our approach here and a big acceleration of this program. We have a broader pipeline of indications coming through, and our second program is MT1980, which is a candidate aimed at neuroinflammation. Neuroinflammation is, of course, a very big and expensive problem that precipitates or accelerates a number of brain disorders, including things like Alzheimer's disease, and it's, of course, key in areas like stroke and traumatic brain injury. We're interested in demonstrating proof of concept for our biomarker and drug as quickly as possible. And so for that reason, we're targeting initially a very acute form of neuroinflammation, which is called post-operative cognitive decline, or POCD. POCD is essentially a permanent cognitive impairment caused by inflammation from a surgical wound anywhere in the body, compromising the blood-brain barrier and allowing inflammation into the brain. This is a very expensive issue because a very large number of elderly individuals undergo surgeries every year. So for each of the over 65s who takes an elective surgery, think of all the knee replacements and hip replacements, for example, unfortunately, there's about a 15% risk with each of those surgeries of developing POCD. There are no drugs approved. There's no way of knowing who's at high risk for this at present. And so Monument has been developing a combination of a risk biomarker to detect who is most at risk and a drug to treat those most at risk, which I'll tell you about now. The biomarker that we're using here would be completed prior to surgery, taking about 10 minutes at a pre-surgical visit. And essentially, it's measuring specific aspects of memory that tell us how healthy the brain is in the individual prior to undergoing the surgical procedure. It allows us to stratify patients into those who are at high risk of POCD because their brain suggests that they already have some early stages of some neurodegenerative condition, versus those who are at normal risk. For those who are at high risk, we're proposing to treat them with a small molecule with anti-inflammatory effects that has been reformulated in order to get it into the brain. We have strong preclinical data showing that we can penetrate the brain with a small molecule and have a strong anti-inflammatory effect in the brain when we do that. And our first phase 1 data also shows good brain penetrance and safety. So we see a linear relationship between how much of the reformulated drug we dose orally and the amount of drug that we're able to detect in the CSF. So we think here we have a good candidate for treating neuroinflammation. We could use this prophylactically in those at high risk of POCD and also potentially symptomatically in POCD and other conditions. The next steps in this program are to start a clinical proof of mechanism study in healthy volunteers, and we're also validating the biomarker and tweaking the algorithm to make it optimally sensitive in conditions like long COVID and chemo brain, so brain fog associated with chemotherapy. We think that these are other conditions where the ability to recognize who is at high risk or currently experiencing cognitive dysfunction because of neuroinflammation and then treat with this anti-neuroinflammatory drug is a potentially extremely high value, high impact. So we think that precision medicine is the future of neuroscience, and we're not alone in saying this. I'm showing you now some other companies working in the area with a wide variety of technologies. What we have in common with them really is that we are trying to better develop drugs by treating the brain basis of disorder rather than the clinical diagnostic categories that are useful in clinical practice but have not historically helped with drug development all that much. These companies have a range of candidates with pipelines typically involving a number of phase 1 and phase 2 candidates. Their valuations range from tens of millions through to, well, Neumora, for example, who did a public listing recently with a valuation of around $2 billion. So there's a good deal of investment now happening in this area, which is fantastic to see. What we have that's slightly different from these other technologies is this entirely digital platform, the advantage of which is really about scalability, so allowing us to implement this very easily into international clinical trials and ultimately anywhere in the world in clinical practice. These game-like assessments that we use give us the same precise and objective measurement of brain function as other technologies, but in a way that is really easy to use for patients, for sites, and ultimately for doctors. And we hope that this approach will allow us to really improve the rates and success of drug development in CNS in the immediate term, and then ultimately enable the kind of in-clinic decision-making that has taken such a step forward in oncology, for example. We look to areas like oncology and think that this is where neuroscience should be heading, both in terms of the success of drug development and the improvements in outcome for patients as a result. Drug development is, of course, a team sport, so I'd just like to acknowledge the rest of my team, particularly Kiri Granger, Cheryl Caswell and Mark Traherne, who were my co-founders as we spun this company out of Cambridge Cognition in 2021. We've also benefited from a very experienced and kind advisory board, particularly Paula Moran, who has completed a lot of the preclinical work with us, and Robin Bannister and his team, who are responsible for the lipid formulation that has allowed us to get MT1980 into the brain. And on that, I'd just like to thank you for your time and say that if you have any interest in working in this area, please do get in touch. We're always open to collaborations. Thank you very much.
Thank you, Jenny. Lastly, we'll hear from Rosanna Zhang, Vice President of Corporate Development at ACROBiosystems. She previously served as Head of Strategic Initiatives at ACROBiosystems, leading its innovative endeavors across the globe, starting from A-Neuro and transferring it into the company's global seed fund, focusing on investment and licensing in life science tools. Over to you, Rosanna.
Hello, everyone. I'm happy to be with you today to share our perspective on challenges and opportunities in CNS drug development, and especially how new tools can enable better preclinical studies. First of all, please allow me to give a brief introduction of ACROBiosystems. Over the past 10-15 years, we have been dedicated in developing better tools to support drug development. Starting in 2010, we have grown on to over 800 people, setting a customer base of 8,000 over 17 countries, working with nearly all the top big pharmas, biotechs, and CROs. We work on facilitating new target screening, CMC establishment, and both preclinical and clinical studies for novel therapeutics. We see a lot of advances in both better understanding of the MOA underlying CNS diseases and the rising of new therapeutic modalities, such as AAVs, where a lot of rare diseases that have a genetic mutation can be cured through gene replacement therapies. We also see a global trend of encouraging CNS research, that the brain-initiated projects in different countries have actually fueled the CNS studies in many aspects. In despite of these advances, the core pain point still exists, which is the high cost and high failure rate of CNS drug development. Because there is a huge gap in translating the medicine from preclinical to actual approval. That is mainly caused by poor efficacy that comes out when it's reaching like phase 2 or phase 3. And for CNS drugs, poor BBB penetration is also a common issue that occurs, as well as the safety issue. So we really need some better biomarkers and models for CNS preclinical study that can really precisely predict the drug's performance on humans. We have been trying to address this issue in the past few years, and traditionally our main products or what our customers are reaching out for are mainly like target proteins, cell lines, and assays. And actually, we have covered the majority of neurological diseases, such as neurodegenerative disease, brain tumors, neurofunctional, neuropsychiatric disorders, and we also had some diagnostic biomarkers and some cytokines for cell cultures, basic tools such like this. But we've been thinking about what is really needed for the improvement of CNS preclinical study. So the general idea is to develop better ways of modeling the disease state and better ways to measure the drug impact. We have summarized 3 main aspects that we can improve. The first of all is the way that we induce the disease. And taking the example of neurodegenerative disease, as we know that they are mostly sporadic, and the pathogenesis of this disease is not that clear till now. And for modeling of this disease, most people would use genetic model or chemical or physical induced model that can capture some of the phenotype of the disease. But this may be not enough to really recapitulate the pathogenesis, the pathology, or the progression of the disease. So a prefrontal model is increasingly used in modeling Parkinson's disease and Alzheimer's disease. And the fibrils that we injected in the animal is able to induce the aggregation of the endogenous proteins that recapitulate the natural pathogenesis and the progression of the disease without modifying the protein itself or modifying the genetic background of the animal. And second, we can improve the system that we use to model the disease. And during many years, we have been using 2D cell lines and animals to model the disease. And there is actually a huge gap because 2D models have their drawbacks. They sometimes are not genetically stable. They lack a systematic aspect of the disease. So more and more people are using organoids and organ-on-chip models that are more physiological, and they use iPSC-derived human cells, so that model can capture a human genetic background, and they also take shorter time to build. And lastly, we can also improve the biomarkers that we use to measure the drug impact. Like when we study neuroscience, we cannot neglect the core property of the neurons, which is their electrical activity. However, most of the biomarkers that we use for CNS drugs are biochemical markers. Only in a few cases, such as measuring epilepsy, E-phase biomarkers are used. But the thing is, in many other diseases, such as movement disorders or dementia, electrical differences between patients and healthy people have been reported in many papers, but they are not really applied in the drug development. Having said what we can improve, in the next parts, I would like to introduce the tools that we developed here in ACRO that we hope to contribute to the improvement. First, we have developed prefrontal models for AD and PD. And as we know that protein aggregation is a major pathological feature of neurodegenerative disease, and this aggregation process can be simulated by introducing a fabricated fibril into the system and model neurodegenerative disease. So in brief, the mechanism is that under pathological conditions, pathogenic proteins can integrate into a wide array of undesirable structures, ranging from oligomers to fibrils and to highly ordered aggregates. The fibril structure, the presence of this fibril structure actually represent a rapid growth phase of protein aggregation, as these fibrils are active and can rapidly recruit monomers for elongation. And in addition, these fibrils can also randomly break into shorter fragments that could act as seeds, which can transmit between cells and can recruit monomers independently. So preformed fibrils are in vitrally fabricated fibrils that have this seeding activity. By introducing these fibrils into our cell culture or into the animals, they can recruit soluble endogenous pathological proteins to form aggregates and finally induce neurodegeneration and capture the pathology of the disease. So why PFF models are increasingly employed, what they can do. So here is a comparison of PFF models and genetic models, taking the example of alpha-synuclein model for Parkinson's disease. So basically, these two types of models can simulate different process of protein aggregation and degradation. So they can answer distinct scientific questions and test different therapeutic approaches. As we can see here in the table, genetic models, they mainly model the degradation process. So they are perfect to test therapeutic approaches such as antibody drugs that aims to eliminate protein aggregates. But PFF models, they can better recapitulate the transmission and uptake of the pathogenic proteins, which is important to study the disease propagation. So PFF models is essential for the development of some early interventions or some disease-modifying therapies. People may ask that can PFF models really recapitulate the phenotype of the disease? Actually, a lot of labs have done this kind of experiments and demonstrated that PFF injections can induce disease phenotype in wild-type animals, especially with alpha-synuclein PFFs. The injection can induce all the typical phenotypes, including alpha-synuclein inclusion, Lewy body formation, dopaminergic neuron degradation, and motor and cognition defects. But to obtain a significant and consistent phenotype, we need to use really high-quality PFFs with high purity and high bioactivity. In ACRO, we ensure our PFFs to have the maximized bioactivity by strict quality control. We use high purity protein monomers to fabricate the fibrils. As you can see here by the SDS page, all of our monomers have greater purity of greater than 90%. And we control the fibrils bioactivity by 3 methods. First, we do a THT control. We have to see a massive increase of the THT signal, which indicates the beta sheet structures in the proteins. And then we verify the structure of the fibrils by microscopy, and we have to see this consistent structure of fibrils. Then we also do a cell-based assay that we seed the fibrils into our GMP labeled alpha-synuclein cell line and to see whether these fibrils are capable to induce the aggregation of the endogenous alpha-synuclein. Data of our cell-based assay, the first row is the control, the second row is the positive control that we see the cell lines with the monomers. As we can see, both the negative and positive control shows no significant green fluorescent spot. And the third row is the seeding assay of our PFF. We can observe the clear green light spot here. And the last row is the same experiment using our competitors' PFF. So you can see that our products have the better bioactivity. We are developing better ways to induce the disease. They have to be based on a system that actually human-like. With the push from the FDA at the Modernization Action 2.0, we have already seen the regularizations is embracing the alternative disease models compared to animal model, and encouraging the use of organoids and organelle chips. And the organoids are mainly derived from human iPS cells, so compared to animal models, they can better capture the human genetic background. And usually, organoids have a 3D structure and contains more than 2 types of cells. So as a model compared to 2D cell cultures, they are more physiological. But organoid application is still under maturation, with certain pain points need to be solved. For example, they have limited vascularization and is not easy to model immune system with organoids now. But regarding the pain points, ARCO have developed other solutions. Well, from the technical aspect, there is not a standardized protocol for growing organoids. Different companies and labs use different methods, which may lead to inconsistency results while testing drugs. And usually the protocols of growing organoids includes many steps of medium change that may introduce as well variabilities in the drug testing. In addition, there's no standardized standard QC requirements given by the authorities. So to obtain trustable results of drug development, a simplified protocol, standardized QC is really needed. And another thing is that organoids often take long time to mature, but with long-time culture, organoids grows bigger and bigger, which may cause nutrition shortage for the cells in the middle of the organoids and cause cell death. Many will add matrigel in the culture system to ensure the long-term culture, but matrigel may sometimes hinder the electrophysiological recording, which is a big issue for brain organoids and heart organoids. So we really need an accelerated maturation technique for organoids and the capability to grow vascular, maybe inside the organoids, to ensure long-term culture. And secondly, organoids are very criticized about their environment, so making them difficult to be transported. So for better commercialization and allow more people to use this technique, we have developed a transport buffer that enable the fresh transport of organoids, as well as a cryopreservation solution to enable the long-term storage of organoids. And finally, to make organoids a good model, we need to develop some associated assays. Since the immune system is crucial for many diseases, so the capability of build immune system associated with the organoid is also very important. And for organoids that have electrical properties, such as brain organoids and heart organoids, a 3D recording system is needed to really capture the 3D activity of the organoids, not 2D recording. Are the 2 types of organoids that we already developed, the cardiac organoids and cerebral organoids. We can offer fresh organoids as well as the differentiation kit. And our advantage is that we have our cryopreservation solution, so you can store the organoids for long term. We have a simplified protocol, high batch consistency, and for brain organoid, we can grow vascularization brain organoids, and we don't need matrigel for long-term culturing. And we are now developing more organoids, liver, retina, other brain regions, and we are also developing a high-throughput organoid recording solution. I mentioned here are 2 examples of the assays that we are developing now. The first one is organoid-based PFF models. So the way we induce the disease is using preformed fibrils, and the system we use is the organoids. And adding these fibrils to the organoids, we will observe the phenotype of the disease, and then add the candidate compounds to the organoids. We can have certain these kind of readouts, such as cell counting the live cell, measuring the phosphorylation of the proteins, measuring the secretion of cytokines, visualize the protein aggregates, and even measure the electrophysiology activity of the organoids. And the second one is assay that I mentioned. It's a high throughput organoid recording assay. We are going to have this kind of instrument that allowed automatic high throughput 3D recording of organoids. This will be fully automatic and measure compatible and really capture the in-depth activity of the organoids. To summarize, here is the whole picture of ACROBiosystems' vision in the field of neuroscience. We aim to provide a total solution that serves different stages of CNS drug development. We have proteins, preformed fibrils, a modeling tool. We have antibodies, stable cell lines. We have a total iPSC solutions, including basal media, supplements, antibody differentiation kits. We have a total organoid solution, including fresh or cryopreserved organoids for heart, brain, liver, intestine, retina, etc. And we are also developing in vivo and in vitro assays, as well as electrophysiology solutions together with our partner, Diagnostic Biochips. Thank you everyone for listening. This is our honor to share our observations at DDW. Please feel free to reach out for any question or partnerships. And I'm happy to answer some Q&As right now.
Thank you, Rosanna, and thanks again to our other speakers for those fantastic presentations. Shortly, our speakers will take part in our Q&A, but before that, I'd just like to remind everyone that this webinar will be available to access on demand on the DDW website, www.ddw-online.com. Okay, now we'll move on to the questions. Right, Fiona, a question I have for you here is, how could a biomarker-based blood test for sensitive detection of Alzheimer's disease pathology, specifically measuring P217 tau, potentially revolutionize disease research by simplifying the process of identifying suitable patients for clinical trials?
Thanks, and that's a great question. I'm going to answer this specifically targeting those preclinical patients who've got the Alzheimer's changes in their brains, but they don't know it yet, and they don't know that they're going to progress to Alzheimer's disease within the next 1, 2, 3 years. So for those patients, how do we enroll them in our clinical trials? Previously, when they came through the door, they were interested through, reasons, personal reasons to be involved in Alzheimer's research and drug development. Previously, we'd either have to do invasive lumbar punctures to measure CSF proteins, or run them through multiple PET scans to identify specific levels of the Alzheimer's disease proteins in their brain. And you can imagine going from cognitively normal but concerned people to actually identifying the subset who've got Alzheimer's disease changes in their brains. You're going to have to screen a huge number of people in order to identify and enroll the right patients in your clinical trial. With blood tests like this plasma 217 plus tau assay, we're able to insert that blood test into that pre-screening funnel. So a potential patient or subject comes in, they're interested in the clinical trial, rather than saying, right, now we're going to schedule you for your lumbar puncture, we're going to schedule the operating room, we're going to schedule the MRI suite and the tau PET. Patients can simply have a vial of blood taken, screened for this levels of plasma 217 in their blood, and then identified as to whether they would be cases for that confirmatory screening going forward. So you can see that especially in this preclinical space, that's going to hugely impact the success rate of really identifying the right patients to enroll in our clinical trials going forward.
Thank you. And another one for you, Fiona. As biomarker technology continues to advance, how will this affect the development of patient treatments in the future?
Thanks, that's a great question. I think that in the future, we're going to have really convenient biomarkers that are going to be able to identify patients who have changes in their brain, who are at risk of developing and progressing to cognitive decline, and even where we could identify potentially subtypes of Alzheimer's disease or comorbidities of Alzheimer's disease. So, I foresee that actually biomarker tests for Alzheimer's disease are going to be offered to patients in the clinician's office side by side with measuring their LDL and HDL and A1C levels to really identify patients who are early in the Alzheimer's cascade and even before that they've really progressed to cognitive decline. Catching patients early, identifying the right patients for the right therapy, and then being able to direct them to meaningful disease modification, disease slowing treatments, I think is really going to be able to help us keep those patients in very early stages of disease where they're active, they're engaged, they can communicate with their families and live independent lives.
Jenny, the first question I have here for you is, will you take these drugs all the way to market or partner for late-stage development?
Yes, we absolutely plan to partner for late-stage development. So for our schizophrenia program, for example, the data should be available in 2026, and we'll be hoping to partner from that point for phases 2 and 3. We're also interested in working with companies where our digital biomarker might help their drug to find a better target or have a better effect in a specific patient group. So absolutely, we're a small company and we're looking to partner and collaborate to expand our impact.
Another one, Jenny, how widely applicable do you think digital biomarkers are in neurology and psychiatry?
I think digital biomarkers are extremely widely applicable, particularly in neurology and psychiatry, because these are areas where diagnosis and a doctor's understanding of what's going on is basically derived from question and answer plus what they can observe of the patient themselves. So the idea with digital biomarkers is if we can measure something objectively and preferably measure it repeatedly and non-invasively and at scale, then we can really begin to understand what's going on for this patient, not rely on their memory and their insight into how they're feeling, and allow a sort of pattern matching process, which is what psychiatrists and neurologists do, to be based on much more data than they currently have available.
Thanks, Jenny. And lastly, what do regulators think of this kind of approach?
So regulators are becoming much more comfortable with digital biomarkers in general, I would say. They are used to seeing these specific digital biomarkers as the endpoints in clinical trials. That's how Cambridge Cognition uses them. So very comfortable with that. They've also seen them in medical devices, etc. So I think, digital biomarkers are becoming much more common in clinical trials and drug development in general. And the idea of pairing a biomarker with a drug is, of course, not new. That's been going on in oncology for a long time. So regulators have a specific combination pathway that deals with that.
The first question I have for you here, Rosanna, is in your preformed fibrils products, are they all fibrils?
Well, for this question, actually in our preformed fibril products, the fibrils and the monomers coexist. But we control the portion of the fibrils at least 50% to ensure the bioactivity.
Thank you. The next question for you, Rosanna. Do your cardiac organoids show regular beats?
Yes, our cardiac organoids show regular beats from about 12 days of the culture. The beat rate is at about 30 beats per minute.
Thanks for that answer. The last question I have for you, Rosanna, you mentioned a high throughput organoid recording assay. Can you talk a little bit more about this?
Yes, we are working on an automatic and high-throughput organoid recording solution. Because during our research on brain and cardiac organoid, we found that current organoid recording with MEA or patch clamp have certain limitations. For example, if you are using Matrigel while growing the organoid, then probably you won't get a good electrical signal. And while using MEA, we have to grow the organoid on the plate for like about 2 weeks so the cell can be attached on the plate. And while using patch, many usually have to dissociate the tissue or slice the organoids. So with this method, we question whether the in-depth signal can really be captured. Do we really have a 3D signal? Then we actually tried to record organoid with silicon probes using an in vivo electrophysiology method, and that gave us a much better and much significant results. So to adapt the requirements of drug screening, we would like to turn this recording method into an automatic and high throughput platform, which will allow long-term growing and automated recording of at least 96 organoids.
Thank you very much. That brings us to the end of this webinar. I'd like to thank everyone for tuning in, and a big thanks to our presenters. If you would like to listen to this webinar again, it will be available on demand at www.ddw-online.com. Thank you to the sponsor of today's webinar, ACROBiosystems. You can now view Drug Discovery World content online on your mobile or tablet or via the DDW app, which can be downloaded via the App Store or Google Play. Thanks for listening.